the smoking process and why it leads to a unique feeling for the consumer. We have international patent pending technology to extract the compounds from smoke and formulate it into a product for consumers to enjoy. We recently released a product that is a hemp derived smoke extract. It is completely legal to purchase in any state, and if you would like to try some, please find us at www.profoundnaturals.com. That's www.profoundnaturals.com.
And for all of you who sent us feedback, thank you so much. We really, really enjoy it. Before we get started, I have to apologize for my audio quality. This was our first run on a new platform, and it will be better in the future. So I'm really sorry. But all right, let's get right to it.
Welcome to Smokin' Science. Nothing but the facts about our favorite plant. Okay, so thank you all for coming to our first ever free webinar from Team Smokenol. We're really stoked to be doing this for the first time ever because it's been a dream of ours to continue producing high quality content for free for everyone. This talk is going to be on the pharmacology of cannabis smoke. And just before we begin, I'll just do a quick introduction of myself. So my name is Dr. Riley Kirk. I have my PhD in pharmaceutical sciences and I specialize in natural product chemistry. So I study the molecules that plants, bacteria, and fungi produce and the ways that we can use these chemicals as drugs or drug-like compounds. I met Dr. Shields about a year ago, almost to this day, and we had very complementary thoughts on how nature could be used as medicine, how education can work in the 21st century with the new technology, and what our target audience is. So we're really excited for things to start coming to fruition and offering things like this.
And I want to just say that what we do, and actually not even what we do, but like Riley and I meeting each other. Okay, hold on, mate. Let me take a step back. Hi, I'm Dr. Miyavi Shields. I have my PhD in pharmaceutical sciences as well. I did my PhD studying the endocannabinoid system. I studied the system in the brain and the body that interacts with cannabis. I did like atomic level interaction of the molecules, and you all, if you have seen me before, probably have seen me with this. And I studied how these molecules interact in the brain and body and then how it causes the effect. And what I wanted to say, just to thank all of you for being here. I want to thank you for being here because this is monumental for both of us. This is our first formal webinar that we're giving together. And as Riley said, we met about a year ago, had a lot of things in common. And we would not have met if it weren't for all of you. We don't know what the universe ever has in store for us. We just are in the universe and doing things. And I was in the universe, being like, I'll start educating about cannabis. And actually, I had this model. This isn't the original model that I had. I didn't have a molecular set. I actually only had THC. And I had it as a picture. It was like on my wall. And so I pulled it down and I was like, I'll just make a quick little video with this and post it on TikTok. And everyone was so interested in the science and so passionate. And people started tagging us together. And that's how I found Riley. Riley was the second video that I ever saw on TikTok. And I've been in the endocannabinoid eicosanoid field. I've been researching the molecular pharmacology of these molecules for 12 years now. And I've never come across someone like Riley. And so when we met up in real life and we're like, whoa, we should definitely work together. This was something that we talked about that I was really excited about, which was creating high quality, high intent, density. We're going to go pretty deep here, but we're going to do it in a way that we hope we can have everyone understand. So I'm going to get started with the very, very beginning. which is, what is pharmacology? So both Riley and I have our PhDs in pharmaceutical sciences. That means we studied pharmacology. Pharmacology is the study of drugs. So there are two different ways that we can study drugs. Normally, pharmacology is split between what is called pharmacodynamics, which is how the molecules interact with the body, or pharmacokinetics, which is like how quickly they do it, how fast they're absorbed, their metabolism in the body, like how long they take. If you think about like dosing, like timelines and dose schedules, that's pharmacokinetics. We are going to be mostly talking about pharmacodynamics in this lecture, which is what these molecules do and how they cause an interaction. What you are looking at in the video on the right is the CB1 receptor interacting with THC, and I will go into more detail of that later. But first, I'm going to cover what is a drug. So, a drug is technically any molecule that comes from outside the body to cause an effect. So in cannabis, the active molecules, which Riley will go over in a little bit, come from the plant. We smoke it, we eat it, we inhale it. It comes from outside the body, and we take them, and it causes an effect. This is what makes it a drug. The definition of a drug is really, really broad, and it's really, really, it's variable depending on who you talk to, right? Because like some people consider certain nutrients to be drugs, certain, poison, medicine, toxin, nutrient. All of these are things that technically fall under the category of drug. So what is a molecule? A molecule is any group of more than one atom. So if you're still looking at my video of me, here is THC. This is delta 9 THC, and it's a molecule. The reason why it's a molecule is because it is a group of atoms. Every single one of these little circles here, these black circles, and then these red circles are atoms.
Okay.
Oh, am I good?
I thought you froze for a second, but I see you moving again, so it seems to be
hear
okay.
me? Cool.
I can hear you.
So these atoms are held together by bonds. That's what these little gray ones here are, single bonds. The purple ones are double bonds. And when you have atoms that are held together by bonds, any group of more than one atom is a molecule. So now, last question is, what is an atom? An atom is the basis of absolutely everything physical in the universe. And an atom is basically, you can have different atoms. There are different types of atoms, right? This is a carbon atom. This is an oxygen atom. And when you put those different atoms together, they make different things. And so essentially for pharmacology, all we have to know is that these drugs are a group of atoms, and they're different shapes, and they're different structures. So here's another picture of delta 9 THC. This is probably the most common one you've seen. It's like a stick figure depiction of it. And it looks a little bit different from what I have here. This is the way that it looks on paper. This is not the way that it looks in real life. Because if you look at my model right here, I'll try and get out of the way so you can see it on a cleaner background. It's flexible. The molecule can rotate. It can move. And actually, in the case of CBD versus THC, that's one of the big differences, is how flexible the molecule is. And so if you look at the video on the very far right, THC is in a specific pose. It's shaped a certain way when it interacts with the receptor. And that's what we're going to be talking about later, which is mostly how did the changes in shapes change like what happens on the receptor, which is what Riley's going to cover next.
Yeah. Also, I just want to make a note about molecules, because it seems kind of like a crazy concept when we think about it. But I know a lot of people on my TikTok and other platforms have been a little confused about the scale of a molecule. Thinking that you could see a molecule with something like a microscope, but we don't use microscopes to look at molecules. But if you have a bunch of molecules that are the same molecule together, you might visualize it as like a white powder or a crystal. So that's when a bunch, like millions of molecules come together. You can visualize it in that way, but we have to use very sensitive instruments otherwise to study molecules and these special interactions.
So I just want to quickly say that not everyone, but a gram is probably like a certain amount that people I think about, right? Like we think about a gram and we say, okay, I can visualize that, like a gram of isolate in here. How many molecules is that? Okay, so if it were 1,000,000 molecules, it would be 10 to the 9th power. But it's not 1,000,000. It's far more than that. It's 10 to the 21st power. So it's actually a million molecules. So that's just a context of like, if you had a gram of isolate, it's like a million times a million molecules of what's in there. They are very, very small. We are talking, we cannot yet see them with microscopes. Sorry, that's like microscopes. We have to see them using x-rays, which is how this video on the right was made. But okay, that's not today's topic.
Okay, so when we're talking about, we're going to talk a lot about receptors today. Receptors are located throughout our entire body. We often use what we call like the lock and key analogy when we're talking about receptors. This is crazy oversimplified, but it's a good way to get the thought in your head of what receptors are doing. So when we think of this lock and key analogy, the lock is going to be the receptor and the key is the drug. Okay, so if the drug fits into that receptor in a perfect way and can activate it, it could unlock that receptor, which is going to cause a bunch of changes in your body and maybe makes you feel high or maybe reduces inflammation or something like that. But we can also inactivate receptors as well. Maybe that key kind of fits in and locks it instead of unlocks it. Or maybe the key kind of sticks to the top of the lock so we're no longer able to unlock it. So there's many ways that key can interact with that receptor. But I think the really, really cool thing to think about with receptors is we often think it's like the drug that's causing all these crazy things happening in our body. But the receptors are already in our body. The keys are just allowing us to feel certain things. So when we think about receptors, we have receptors on every cell in our body. And if the right drug can interact with that right receptor or drugs interacting with different receptors, the pharmacology can get really, really interesting. So maybe it's not just interacting with one receptor in one system of our body. Maybe it's interacting, or different compounds are interacting with a bunch of different receptors in different systems in our body. And together that's making us feel a really unique effect. So when we think about it, lock and key, but it is much more complicated than that. We're also going to kind of touch on enzymes as well. And just like receptors, enzymes are already in our body. So when we find we have keys or something that can activate enzymes as well, we also have things that can deactivate enzymes. But enzymes in general are like these little machines. They always have a job, whether that is to translate and transcribe DNA to actually make proteins, or whether it is to help compounds transport across membranes. Those enzymes always have a very specific job, and they just do their job all day long. So we'll say that they help catalyze reactions, because really we wouldn't have much going on in our body if it weren't for enzymes helping everything move along in the right way.
And I think really quickly before we move on, I want to emphasize agonist and antagonist, because we're going to come back to these words in a couple slides and then again at the end as well. Agonist loosely meaning like turning on, and antagonist loosely meaning like not turning off but blocking the ability for something else to come turn the receptor on. These are two of the main states of how drugs can interact with receptors, right? It's agonist or antagonist. This is like a yes or no. It's a binary, but that does not describe what we know. What we know so far about receptors and how drugs interact with receptors is that is very much so a spectrum. And like many things in this universe, that we as humans can understand it and conceptualize it better as a binary of like on or off, agonist or antagonist. The reality is that there's a huge spectrum or continuum of how these molecules change and interact with these receptors. So I am going to quickly go over the very, very basics of molecular mechanisms for both receptors and enzymes and what that means. So here we have the drug, let's call it like THC. It is the little orange thing, and here's the receptor. The receptors are like these little cylindrical things or whatever, and when THC binds the receptor, you might notice that there is a change of shape. Some of those cylinders look different. That's because when it comes in and it sticks to that receptor, it's sticky in certain areas, and then it's sticky and it has like Velcro that's that's very specific to the receptor, and it pulls the structure of the receptor into a different location. And that change in shape is what causes the activation of these receptors. It is called a change of conformation. So it's the conformational change of the receptor that is what causes it to be activated. So when these molecules are floating around in the body, when they are interacting with these receptors, they are actually, I would say physically, but it's chemically as well, but they are changing the shape and structure, and that causes a signaling cascade that is very, very complicated and perhaps will be its own webinar. But these changes are what causes the effects. So the drug comes, changes the shape of the receptor, that activates the receptor, deactivates the receptor. It causes a signaling cascade, and that leads to the effects that you feel. Now, for enzymes, Riley mentioned they are little machines. In this enzyme picture, my little machine is taking diamonds and turning them into triangles. That's its job. And if in the case of the endocannabinoid enzymes, it would be taking anandamide and turning it into arachidonic acid and arachidonic acid and ethynylamide. But these enzymes are just doing these jobs. This is its specific job. And if you were to treat it with a drug that blocks its ability to do that, then all of a sudden you're going to have a whole lot of diamonds and no triangles. And this will lead to effects. And I just want to point out really quickly before we move on that the endocannabinoid enzymes, and I also have to disclose that I'm biased because that was my focus for my PhD, the endocannabinoid system enzymes are so important. They are so often overlooked. We talk a lot about the CB1 receptors, but the endocannabinoid enzymes exist as well, and there is no way that that they don't have and that they are unaltered in cannabis.
Yeah, and just to put that into context as well, so when we talk about the endocannabinoid system enzymes, the reason we're highly focusing on these enzymes, just in general, what research is kind of going towards as well, is because the endocannabinoid system, when it's making these compounds like AEA and 2AG, it's producing them on the spot. So when your body needs them, it's going to synthesize them on the spot. That's an enzyme that's synthesizing that. It's going to go act on your cannabinoid receptors, and then a different enzyme is going to break it down and recycle it and then use them again. So because it's happening in such a localized place and it's producing the compounds that interact with our endocannabinoid system, those enzymes are really important. And if we want to introduce more compounds to interact with the endocannabinoid system, we can either introduce phytocannabinoids like the ones from the plant, or we could manipulate those enzymes to maybe not break down the endocannabinoids as quickly. So they're kind of sticking around for longer, and they could have additional therapeutic effects that aren't from the plant. They're actually just leveraging our own system.
That is such a perfect transition into my next topic, which is the pharmacology of CBD, or the lack of a clear pharmacology of cannabidiol or CBD. Because some people believe that one of the reasons why CBD is so helpful for epilepsy has to do with its interaction with the other medications, that it would potentiate it via what Riley was talking about by allowing the other medications to be around for longer or be stronger. But I'm going to talk about the endocannabinoid system targets of CBD really quickly. And it's complicated. CBD has over 65 potential targets in the body, and that list is definitely not complete, in my opinion. And the reason why I'm bringing this up is because the theme of our talk will come up. We have a theme going here, and the theme is going to be about this unknown potential. And I think that one thing we have to keep in mind when we look at pharmacology, specifically the level that we're talking about, which is receptors interacting with the molecular structures of what's in the plant and what's in the smoke, is we have to keep in mind how we get this information and what does that information actually mean. Okay, so let's go through the endocannabinoid system targets. CBD has been shown to be an antagonist at the CB1 receptor at high concentrations. That means that at high concentrations, it sticks to the top of the receptor and it doesn't allow anything else to bind. It doesn't allow THC to bind. This is up for debate still because it was done earlier, and it needs to be repeated like almost everything. And it gets really complicated because other people found CBD to be something called a negative allosteric modulator. That is a fancy way for saying that CBD turns down the dimmer switch on the CB1 receptor or the volume. It turns it down. So it doesn't completely block anything else from being there, but it'll turn down the volume of that signaling. All right, so that's CB1. CB2 has been shown to be an inverse agonist. This is only at really, really high concentrations and is also still up for debate. An inverse agonist is exactly what it sounds like. If an agonist turns the receptor on, an inverse agonist will turn the receptor off. And that's different from an antagonist, which just sits there. So again, though, it's the more important thing is it reduces it. So for both CB1 and CB2 receptors, CBD will decrease its activity. GPR55 is, some are starting to call it the CB3. I think we should just call it CB3. Like, why not? It also is an antagonist. It decreases the activity there. GPR18 is a more recent discovery one. Maybe CB4, who knows? It's an agonist, so it turns this one on. By the way, GPRs, they're what's called, they call them orphan G proteins. And these are receptors that we know exist in the body, but we have no idea what they actually do. And so we just give them a number. That's a part of my next slide, so I'll come back to it. But so now we see that it's getting kind of complicated. It's complicated here because we have 3 of these receptors that CBD is turning down, right? And then we have one receptor that's turning up. And then now we have this receptor, the TRPV1 receptor. This happens to be the receptor if you like spicy food and capsaicin specifically, so like chili peppers, any hot pepper. This is the same receptor that interacts and causes that heat in your mouth. CBD is an agonist or turns on this receptor, but it gets complicated too because this receptor specifically has a mechanism where when it's turned on too much will actually just disappear and it decreases, which is why people who like spicy food a lot can tolerate a lot of spicy food because you literally have a tolerance, and this is part of the reason why. So this is starting to get complicated, and now we have Pha. Pha is an enzyme. It is a metabolic enzyme, which means that it takes anandamide and turns it off by metabolizing it. So if you inhibit it, right, you end up with a lot of anandamide. So if you have an increase of anandamide, all of a sudden you're increasing all of these receptors because anandamide is the endogenous within the body, the thing that we make. It is the natural endogenous ligand for all of these receptors. So you're looking at this slide and you're like, what the heck does all of this mean? This is like incredibly complicated, and like, what is going on here? And what is going on here is all of it. All of it is going on at the same time. All of it is going on like right now in my body. And I think that that's incredible. Oh, sorry.
Can you go back?
Yeah.
One more thing you mentioned that I think is really important to talk about is you mentioned dose in a lot of these cases. So part of the reason studying cannabis pharmacology is so difficult is we're all taking different doses of different things at different times of the day. It's all dependent on what flower we're using, what product we're using. Is it really, really high in CBD? Does it have no CBD? Does it have a little bit of CBD? These changes are going to affect the molecular dynamics inside our body. So when we talk about cannabinoids being biphasic compounds, where at low doses they cause some certain effect in the body, and then at high doses they cause a completely different effect, it's really this that we're talking about. We're talking about certain receptors being activated or deactivated at certain doses that's completely dependent on the compound. And we are rarely just taking in one cannabinoid. We're usually taking in maybe dozens. And if they're all acting in this biphasic manner, it really is an unlimited potential of how different people are going to feel when taking in these products.
Absolutely. And then I'm drinking coffee right now. So I would argue that it's almost no one is only taking the cannabinoids. But this really encompasses the theme of what we're going to be talking about for the rest. We have 2 more sections on this. It's the unlimited unknown potential of cannabis. Cannabis smoke is the topic of this one, but cannabis in general, natural products in general. And actually something that we talk about a lot is chemodiversity, which will also come up. But this unknown unlimited potential is exciting. And it's something that we are still in the beginning. We know a lot. We understand a lot, but we also don't. So what you're looking at in this video, which I will have it replay now. Oh, it's going to replay on its own. This video is the enzyme that I did my PhD on, and that's CBD. The black molecule inside of there is CBD. And there's not any publications that say that CBD works on this enzyme because it's never been done. And the research just hasn't happened yet, to my knowledge. I think someone should do it. Please go do it. And the point I'm trying to make here is that there's so much that we don't know. We don't fully understand all of the targets in the human body that cause the beneficial effects. The protein that I did, this enzyme that we're looking at right now, that I did my PhD on, wasn't even discovered until the 2000s, and we didn't know what it worked upon. It has a completely unsexy name. It's ABHD6 because, again, it's similar to the GPRs. When we don't know what something does, we just like give it a number, and then we're like, oh, we'll figure out what it does later. And there are hundreds upon hundreds upon hundreds, thousands upon thousands upon thousands of things inside the human body that we still don't know really what they do. And we don't know whether or not they have any beneficial effect linked to them because we still don't know about them. The next thing is that we don't understand how these things work together. We understand what we know. There's a bunch of unknown that we still don't understand. But even from what we do know, we don't really understand yet how they're talking to one another at the molecular level. So my enzyme, again, I'm going back to this, ABHD6, it is an enzyme that's an endocannabinoid enzyme. And yet it's been linked to NMDA receptors, right? So it's linked to this different system inherently. It controls their expression levels. It controls, so this endocannabinoid enzyme controls the expression levels of a different system. And that's very, very common amongst other things. Like, and it's not just the endocannabinoid system, but it's everywhere in the endocannabinoid system where there's a crosstalk between all of the systems. And we are in the infancy of understanding what does that crosstalk mean? And what does it mean when you change one thing and then it causes the crosstalk to change as well? And, you know, we're not even close to figuring it out. I think it's beautiful and I think it's complicated and it's amazing. But this is a potential that we have for understanding how our body works. You know, we are in the very, very beginning stages of it. And it's something that I'm really excited about.
When we talk about that, we're not even close. Part of this issue is when we've studied traditional drugs and pharmacology, we look at it in a very linear fashion. We say this drug affects this receptor, this causes this effect with minimal side effects. And we really just focus on that linear system. But with cannabis, we have a biological system, the cannabis plant, interacting with a biological system, which is our bodies. And we know there's an incredible amount of variance in both of those systems. So if a human's endocannabinoid system is different than other human endocannabinoid systems, then even if you were to give them the same plant, they might react differently to that. So when we say we're not even close to finding this, the tools we've used to study pharmacology have been very different than the tools that we would use to study cannabis medicine, because we're usually focusing on one single compound at one single receptor. We're not studying 400 different compounds at a dozen or 24 different receptors and what that could mean. Even if we add in genetics, that could, that adds a whole nother wrench into the system. So we're not even close because we don't study things in systems and networks. We study them incredibly linearly. I know that systems are advancing, tools are advancing in science to look at things at a more global approach. And I think that's the most promising method to study cannabis medicine. But currently, that's not the gold standard in understanding drug pharmacology. Okay, I'm going to dive into what secondary metabolites are. So this is what I studied throughout my PhD is secondary metabolites. What I describe secondary metabolites are is the compounds that a plant does not need for survival, but it helps them survive. And I sometimes compare this to humans. If you were to give humans $20. They only have $20 to live off of. They're going to buy shelter. They're going to buy food. They're going to buy water, clothing, that kind of stuff. They need to keep themselves alive. That's like primary metabolites. If that plant only has a certain amount of energy, it's going to use that energy to keep itself alive, to change the energy coming from the sun into energy it can use, to make more plants, to carry on its genes forever. These things that need to be done for that plant to survive, to transport things throughout the body. That takes energy, that takes compounds. Those are primary metabolites. But secondary metabolites are like if you were to give somebody $2,000, that person can now afford all of the food and shelter and water and things that it needs. And now maybe they're going to go out and buy a motorcycle, or maybe they're going to buy an RV or something like fun to experiment with in their life. That's the same thing with the plant. If it has extra energy, it's going to start producing other things that are going to make it more fit for survival. So first it's producing its primary metabolites. That's keeping it alive. Then it's producing secondary metabolites. Those secondary metabolites, which are made in these structures called trichomes in the cannabis plant, they look like little aliens. Those are factories for producing these secondary metabolites that can help protect the plant. So not only are these trichomes physically protecting the plant because they're really sticky, so maybe some bugs or microbes will get stuck on them and not be able to escape, but also it's producing chemicals in these trichomes. These trichomes are these chemicals such as terpenes and cannabinoids are serving a purpose for the plant. Maybe they're helping it protect itself from sunlight so it doesn't get scorched by the sun and die, its tissues dying from too much sunlight. Maybe it's producing compounds that are antimicrobial, so it's preventing microbes from eating the plant and getting energy for the microbe. There's a million different ways. Even smells can both attract and deter compounds. So it's not organisms. So it's using these secondary metabolites in some way that's beneficial to that plant that's going to make its survival easier. So we often talk about cannabinoids, terpenes, flavonoids, these different compounds as being the main secondary metabolites for the plant. But I think it's really important to note that when you're taking in cannabis, when you're smoking flower, you're also smoking the primary metabolites. You are taking in every single compound that living organism is making, and it's going into your living organism. And it's not very simple. It is hundreds of different compounds, and those are going to change based on what flower, what concentrate, whatever product you're using. There's going to be different compounds present, but that plant has made those compounds to interact with other things in the environment in some way.
Also, interestingly, even the exact same clone that's taken from the same mother plant. So cloning is when you take a mother cannabis plant, you have a very clean knife, and you can cut off a cutting, and there'll be stem cells that you can root and then grow a clone. So that clone will be genetically identical to the mother plant, and two clones taking from the same mother plant will be genetically identical to each other. But if you grow those two clones in different conditions, you will get different molecules in those two different plants. And if you take it a step further and you chop down those clones and you trim them, if you dry and cure them differently, you will get different molecules versus the fresh frozen, which is to freeze them immediately. You're going to get different molecules, and that this is part of the magnificence of the cannabis plant, is this giant amount of chemodiversity that you can get. And it's overwhelming to think about it, because we often like to think about it in much more simple terms. But for people who use cannabis and use it regularly, there's a subtlety in the different experiences, and they're distinct from one another. And some of them are very different from one another, and some of them are only slightly different from one another. And it comes from these molecules.
So, as I just mentioned, when we think about using cannabis products, we're often focusing on the secondary metabolites because we think of them as the active compounds in our body. They are the most bioactive. They're bioactive because those compounds can directly bind to receptors in our bodies and facilitate some sort of change that we can feel or measure in some way. But we are taking in all these other compounds that we really don't understand. We think about them as inactive because they're not directly binding to that receptor site, changing the conformation of that receptor and facilitating some sort of change in our body. But maybe we just don't understand this fully because we're still taking in those compounds. So there are other ways that can change the pharmacology within our bodies without directly binding to receptors. One of the ways that it's often published about with secondary metabolites is changes in membrane dynamics. We have membranes over every organ in our body, every cell in our body. We are just a big old pile of membranes that has different things filled in it. Even inside our individual cells, there's membranes in our organelles. There's membranes everywhere. And the cannabinoid receptors and many receptors are located within the membrane of the different cells. So we know that there has been evidence that certain compounds in cannabis can influence the membrane dynamics. If we add through smoking, the actual smoking of cannabis, we know that we're producing some different compounds that change in what we call lipophilicity. So some of those compounds are a little fattier than others, and some are less fatty. And that's really changing the functional group, side chains. If we add carbon atoms, we're adding lipophilicity. And cellular membranes are just really fatty little lipid bilayers. So if we're adding lipophilicity to the compounds, that will likely change the way they interact with the lipids in our body or the membranes in our body. So this could be things like increasing or decreasing the stability of the membrane. If we have less stable membranes, more compounds might be able to get through those membranes. It could also help with like the anchoring of the receptor within that membrane, making it a tighter fit or a looser fit. So there's a lot of changes that are happening when we introduce cannabis, when we use cannabis in our bodies that aren't always directly related to binding to that receptor and causing some change. They could be interacting with the components of our body in unique ways that are not always what we think about when we think about pharmacology.
So the example that I'm going to bring up here is Prozac or fluoxetine, which most people are familiar with. If you're not, it is an SSRI. So we call this molecule, this drug, a selective serotonin reuptake inhibitor, because when it was first discovered, one of its activities is to inhibit this enzyme that reuptakes serotonin. And we called it selective at the time because at the time we believed it was selective. Since then, we found out that Prozac and other SSRIs, part of their antidepressant effects has to do with increasing endocannabinoids. So there's that. But recently, last year, a paper was published that showed that its antidepressant effects were also linked to a different receptor, and that it was specifically linked. It's the TRKB2 receptor, if someone's interested. But it was also specifically linked to the structure of that receptor and how stable the structure was. So when Riley was saying that the changes in the membrane can change the stability of the receptor, it's because if you think about this all as like a jelly, it's moving around. Like in the image, you can see it. It looks kind of wavy. It's moving. It's liquid. We are mostly water. And even though the membranes are fat, they are liquid. So they're moving around, and the receptors are getting kind of like pushed and pulled around with the fats in the membrane. And cholesterol, which is in the membrane, it's in the image here. Cholesterol is something that can help stabilize these receptors. And many of the cannabinoids have been shown to have a similar effect where they can alter the stability of the phospholipids. They alter the stability of the membrane. They alter the stability of the proteins in the membrane. And that if you alter this outside, if we go back to thinking about what causes the effects of drugs, in the very early slides, the effects of drugs are caused by changing the shape of these receptors, by changing the shape of them. And so if we're thinking about something that is changing the membrane, it's going to change and or have an effect on even the ability of that receptor to change shape. And then just one last thing is, what evidence do we have that anything is inactive? I mean, it's just a theoretical question that I'm posing to the universe, is that we call things active and inactive. I have not really seen or come across anything that we put into our bodies that doesn't have an effect on our bodies. So it's just a question posing here about thinking about things that are active and inactive. It's really more of a looking at everything that we're taking as, as a whole. And it actually brings us to this, which is chemodiversity is nature's redundancy. Riley and I, the reason why we're working together is because of this theory. We are developing a theory together. It's the reason why our company does the research that it does, and it's the reason why we believe that there's advantages to natural products that are not fully explored yet. And it has to do with chemodiversity is nature's redundancy, which is really cool because it's a combination of like our thoughts, and I'm going to cover my piece of it, which is chemodiversity. So chemodiversity is a measurement of the total number of different molecules that are present in a product, in a formulation, in an extract, in a flower, in a pharmaceutical product. If I were to take Marinol, a THC isolate, there is no chemodiversity, right? Because there's only one thing. And it's similar to THCA crystals and similar to CBD isolate. And actually, to a certain extent, it's very, very similar to distillate, because distillate that is greater than 80%, distillate that's greater than 60% of just THC or just CBD is not very chemodiverse, right? Because it is almost exclusively like one molecule or one thing versus what's present in the flower. So what we're looking at here on this graph is you're looking at a trace, and every single one of these peaks, these colored little mountaintop peaks that you're looking at, every single one of those is a known and identified cannabinoid. And this is just a sample extract from hemp, so it's from CBD. So the biggest purple one on the right-hand side, that's going to be CBD. But look at all of these other cannabinoids that exist. There are 77 other measurable known cannabinoids in this extract. And we don't often talk about the rares because they're not present in huge amounts in traditional edible extracts, and they're not present in huge amounts in the flower, but they're present when you expose the flower to heat. And that is a piece of what we're going to talk about in the third section, but it's that how does this chemodiversity contribute to the chemodiversity that we have inside us as well? Because this large and just incredible amount and number of molecules that we're taking in, they're having an effect together. None of these things are working by themselves. We're taking them all at the same time. And yeah, that's it.
So, and just to reiterate, like these compounds, although they're really similar in structure, which we're going to continue talking about with nature's redundancy, we see on this HPLC chromatogram that the peaks are really, really close to each other, like in proximity. And that often, not always, means that those compounds are similar in structure, which is why they're kind of grouping together on the HPLC chromatogram. So even though they're similar and they're coming off the column at a similar time, the ways they interact with different receptors in different ways at different doses are completely different. So if we have 78 cannabinoids here that are interacting with our endocannabinoid system in different ways, that really is unlimited potential for the effects that could cause in the body. So just to talk about this a little more, if you've looked at any of the chemical structures of cannabinoids, you might realize that there is a lot of similarities between these different structures. Most of this reason is because when the plants are producing these compounds, they're using certain pathways. So different enzymes are adding different starter material and kind of building these molecules. And maybe a lot of them are the same up until this point. And then certain ones of them are going to branch at this point and branch at this point. So these molecules will be different than these molecules. And then they're going to keep branching and branching, but they'll still have some similarity. We also know that a plant isn't going to just produce one absolute warhead of a compound to fight off microbes. There's a lot of different microbes out in the world, and that plant can't put all of its energy into producing one absolute warhead of a compound. It needs to produce many different ones that might interact with maybe different gram-positive and gram-negative bacteria in different ways, with fungi in different ways, because they have different mechanisms themselves. So that plant is going to produce what I like to call redundancy or molecular redundancy, which often redundancy is a word that we don't want to highlight. Redundant is like you're wasting your time. That's redundant. We've already done that. But nature's redundancy allows for all these different compounds to be produced. And if a certain molecular pathway was shut off, that plant wouldn't just be absolutely destroyed by these things in nature. Instead, it could shift its energy towards one of the other backup molecules or one of the redundant compounds to still survive and fight itself off. You'll almost never see plants producing compounds, like only producing one compound of a chemical class. It's going to produce different ones that are just slightly different to make sure it can still protect itself from many different things in many different ways. It's also important to note that our bodies have redundancy as well. Our bodies have different ways of producing the same thing in case one of us has some sort of genetic issue that allows us to not be able to transcribe a gene, so we can't produce that compound. Instead, maybe we produce it in a different gene in a different location on the chromosome. So again, we're not going to die if something happens. Instead, we just shift our energy towards the pathways that we are able to produce. So it allows for us to be a lot more fit, both the plant and the human, by producing all these different compounds that are interacting in different ways, but might still have the same endpoint, or maybe they have a slightly different endpoint.
And in our bodies, like with the endocannabinoid system specifically, about this redundancy, people know about anandamide, right? Anandamide is an endocannabinoid that our body creates. People might know about 2-AG. 2-AG is the other main or primary endocannabinoid. But there are many, many other molecules that are similar, that are part of the endocannabinoidome. Sometimes they are called elmeric acids. Sometimes they are called endocannabinoid molecules. They are this class of molecules, and you probably can't see what's going on this poster behind me, but I thought maybe we could see it, but we can't. But after the endocannabinoids, there are hundreds. So there are hundreds and hundreds of molecules inside our bodies right now. Every single one of us has hundreds upon hundreds of circulating molecules that are endocannabinoid-like, and it's very, very similar to the plant.
And also, so when you're actually smoking, when you're exposing flower to high heat, transformation is occurring, which is increasing that redundancy, because maybe the compounds we're showing here are the parent compounds, but when they're exposed to high heat, they're changing in small ways, but we're increasing the amount of compounds and we're increasing the redundancy because we have similar compounds that might serve slightly different purposes when they're interacting with our receptors.
And the reason why redundancy it sounds like a negative thing, but the reason why it's a good thing, the reason why nature has evolutionarily conserved redundancy, is because redundancy gives you options. And if you don't have options, then you're shit out of luck. So if you are driving to work and there's only one way for you to get to work, and then a tree falls in front of your path, and you gotta wait for someone to come break down that tree and move it to the side of the road before you could go to work. There's no other option. But if you have 3 or 4 different ways that you could get to work, then all of a sudden that downed tree is not as big of a deal, because you see, oh, the road is closed, I will just go with my other option. And that's why redundancy in nature is important, and why it's a good thing for us to be able to have options. Anything that's alive needs to have multiple options for staying alive.
Okay, so when we talk about this untapped potential, that we have unlimited, unknown, untapped potential, I mentioned that the pharmaceutical approach is very linear. You have this one drug that's in a pretty high concentration that's interacting with maybe a few receptors and causing some effect. But when we have the biological system of the plant interacting with the biological system of us, we need to start thinking in networks, because biological systems never work in a linear fashion. We're always talking about networks. One thing causes a change, which causes 10 more changes, which causes 100 more changes. So when we think about pharmacology of smoking, of flower use, of these complicated plant extracts, whether it's echinacea, whether it's cannabis, no matter what the plant, opium, it doesn't matter the plant. If it's interacting with our body and causing some sort of effect, we need to study these plants in networks. So we have the network of the plant. We have the network of our body. We have the overlapping networks of those plant metabolites interacting with our body. Then we have the compounds our body's making. How do those interact with those same receptor systems by turning off and on those receptors that may have been influenced by that plant? It really is. It is an untapped potential, because we have our own genetics that are going to influence how that plant makes us feel. And even if we had a really good laboratory model, maybe an animal model to study the endocannabinoid system, if we are going to expose cannabis to that animal, that still is not going to be translatable to every single human. We are all genetically diverse, and we will not understand the implications of cannabis medicine and natural product medicine on biological systems until we've studied hundreds of thousands of people in ways that we probably don't even have the technology to study yet. And then maybe eventually we'd be able to link all these concepts together by studying the networks of change that is happening. But right now, I think it's good to just know that there's a lot of unknown and there's a lot of potential, and that we just need to keep developing the tools and working off of each other's work to start understanding what's happening.
And I think that there shouldn't be a goal of saying that we need to absolutely understand everything, because most of research, even in clinical settings, like the gold standard of clinical research on humans, what we're doing when we're doing these studies is we try to get as many people as possible, and then we try to see what happens to the average, right? Like we record what happens to the average person. All of our modern medicine is based upon that. And yet we do also know that no human is average. Not a single one of us is going to be a representation of that average, which is why modern medicine struggles with chronic conditions, because people with chronic conditions are certainly not average, but actually no one is average. And so all of this research and the things that we do, we're limited by the tools that we have. And really, really excited to get to the final portion of this unknown, which is going to throw a little monkey wrench in everything we just talked about, because basically what we've talked about is that it is incredibly complex to look at even just one molecule in the human body, let alone the fact that the plants have hundreds. Now we're going to set it on fire. So heat causes transformation. Greater than 80% of medical cannabis users prefer smoking over edibles. Riley and I updated this statistic to include all of consumers. And with our social media, we got like over 20,000 people to respond. So thank you. If you're one of the people who answered our question, you are part of data. And it was about 76% of all consumers who prefer smoking over edibles. So it's a little bit less, although this other study was way less people, so we don't know. Maybe it's about the same. A majority of people prefer smoking over edibles, and the reason for that is not just about the amount of time or everything. It has to do with the effects. The effects are different. Regular cannabis users find that the effects of edibles to be different than that of smoked cannabis. Now, there are advantages to edibles. They're discreet. You're not super loud and people don't have to know. They last for a long time. And those are huge advantages. Pretty much the big disadvantage of edibles is that 20% of people can't feel them and that they take a long time to hit. So, titration is difficult. But the reason why people prefer smoking doesn't have to do with that initial, maybe a piece of it is that initial onset. Maybe a piece of it is the satisfaction of the oral fixation of smoking. Perhaps. A majority of people I have spoken to prefer its effects. And when we talk about the difference in effects of cannabis, edibles versus smoking, we often hear about absorption. We often hear about metabolism. So we're talking about how the molecules get in the body, or whether or not the liver transforms those molecules before it reaches the brain. What we do not talk about, or what we haven't heard people talking about, is the third thing, which is that smoking cannabis uses temperatures that transforms THC, CBD, and other cannabinoids. We use incredibly high temperatures depending on your method of use. This is also one of the reasons why different forms of smoking feel different. And it's because you are actually creating a different formulation. So what we were describing earlier about how these molecules are interacting with the receptors, with the membrane, with the enzymes. When you have different molecules present, they're competing with one another. They are causing slightly different changes, and little changes in this via transformation can cause really big effects.
So something that I think is helpful for kind of understanding what we're talking about here with these transformations of compounds during the smoking process is we often think of the entourage effect when we're smoking flower or using complex extracts. Maybe it's RSO or something along those lines. But we have these different compound classes. We have terpenes, cannabinoids, flavonoids, all these different compounds dependent on what product you're using that are interacting with your body. And a perfect combination of these different molecules might make you feel absolutely perfect, reduce your symptoms of whatever you're using cannabis for, or just make you feel really good if that's why you use cannabis too. But when we think about smoking and this transformation under heat, under high heat conditions, it's kind of like the entourage effect is extended, because your normal entourage is going to be all those compounds, but when they're exposed to heat, those are going to create derivatives of those compounds and redundancy. And these different newer compounds are also interacting with your body in a specific way. So if you took a strain of cannabis and you smoked it and felt a certain way, and then you wanted to make an extract of that and feel the exact same way, it would likely feel different because you don't have the exact same molecular makeup of your alcohol extract versus your smoked extract. So I think a good way to visualize it is that you're expanding that entourage effect when you're smoking by creating these different compounds that could be just slightly different, but that difference is going to change the way that that compound can bind and interact with receptors. And maybe it changes exactly like the affinity to the receptor. Maybe it doesn't activate it all the way. It only activates it partially. Or maybe it blocks one of these compounds that are produced can block the interaction of a different compound coming and interacting with that receptor. So it really changes the dynamics of what's happening in our body and our brain.
And the things that are on the left of this screen, which is what we're used to talking about with the entourage effect, those things are all present in the flower. And whether or not they're there and in certain ratios, along with things like the moisture content of the flower, the humidity of the air outside, and then the temperature, will all affect what happens on the right and cause those changes. And really quickly, before we move on, if this part is interesting to anyone, and if people are interested in looking into a different extract that is like slightly more similar, that is what we do and that's what we research, and so we should connect professionally, so please reach out to us that way. So raw cannabis has a THCA, the acid derivative of THC. And most people know that you have to decarboxylate that for it to be active. So when you make an edible, you expose it to a low temperature and you decarboxylate it. So there's an arrow to the right there. Then you have THC. What people don't realize is that when you're smoking flower, you are decarboxylating it, yes, but you are also transforming it. So one of those transformations that people may be familiar with is the transformation down to CBN or cannabinol. CBN is associated with sleep, although they still need to be more research done on that. But it's a rare cannabinoid that people are starting to get more and more interested in, right? But there are also other novel cannabinoids present that happen because first, when you expose it to high heat, you get the decarboxylation, and then you start to get more transformation. If anyone has ever taken the roaches at the end of their joints and either smoked them in a bowl or re-rolled them into another joint, it will feel different. And the reason for that is because of this. Because the end or the roach is almost always fully decarboxylated and or partially transformed. So when you go to smoke that, you get more cannabinoid, you get a different profile. It's the reason why resin hits feel different as well, right? And so these unique molecules are what lead to these unique effects because you're taking in this different formulation that interacts.
And also, so like different compound classes, your cannabinoids, your terpenes, your flavonoids, different compound classes have different levels of stability and reactivity. Okay. So when you're lighting them on fire, different compound classes are going to change more than others, which is going to change the ratio of the overall compound classes in your final product. So we know that terpenes are extremely volatile. So after smoking, we know that you're going to lose a lot of the terpenes, but some of those terpenes could also interact with cannabinoids. And in different ways, we can create these different ratios that we don't really think about. But the ratios matter a lot because it really is concentration dependent of how much of those compounds are available to interact with your brain in different ways.
So this is our last slide. Thank you so much, everyone, for sticking with us. And I'm just going to explain really quickly what's going on in this picture before we get into the final unlimited unknown potential of cannabis smoke. This picture is the CB1 receptor, and the red box up there is what we call the orthosteric site. The red box is where THC will be binding. It's where anandamide will be binding. And it's where most things it's where most drugs that interact with the receptor are binding to cause their effects. Now, this receptor is embedded in the membrane, like Riley mentioned earlier, and you'll see in green and in yellow are circled portions of the receptor that are embedded fully within the membrane. And if you look in the yellow circle, you will see that there is a cannabinoid present that is present in cannabis smoke, and that it fits very, very nicely up there against the side of that receptor. So that is something called an allosteric site. I mentioned that word once earlier. It just means it binds to a different location. But what we don't know is how this modification of these different allosteric interactions, how binding to the outside of these receptors will change the way that these receptors function, especially if there's more than one thing bound to it, or if there's something bound to it for a moment and then it's unbound later. What we do know is that it likely causes those changes. And so, like, these molecules that are structurally similar to the parent compounds, the cannabinoids, the molecules in smoke that are very, very similar to THC, very, very similar to, and CBN is a perfect example of that. Another example of that would be cannabis citran, or CBCT, cannabitriol, CBT. These are all very, very similar to the parents, and that they're likely going to contribute through either receptor-specific interaction, which would be the red box, or non-specific interactions, which is the yellow or green.
Yeah, so even circled in the yellow where that compound is binding, that may not ever activate the receptor in any way, but maybe that prevents that receptor from initiating the cascade of events after a different compound acts on it. So the dynamics here, we may be changing things in very subtle ways, but it influences how the more active compounds are interacting with that receptor. It's also important to note some of these compounds that are produced through smoking, or even some of the minor cannabinoids that we don't come across a lot, they could be interacting with these receptors in much stronger ways than any of the more popular cannabinoids. And we often think of it as completely dose dependent, but if a really powerful compound is present in very small amounts, that can still cause profound changes in our bodies. Oh my gosh, we only have 10 minutes left.
What? I thought that it was
talking
supposed to be
forever again.
No, it was supposed to be. Oh, we started it early because we were like, oh, that was our bad. Okay, shoot. Well, we got to finish. Okay. Oh my God, no, I had something else to say. Okay, the dynamics.
Freak out. Sorry.
I'm sorry. That's just the way my brain works. And I'm like, oh no, this is unexpected. Okay. The dynamics are currently beyond our ability to quantify. It's most likely the reason, though, for strain specificity. Because when you're combining these high temperatures with chemodiverse products, you end up with just a giant amount of chemodiversity. And actually, now I remember what I was going to say. There is such a thing called biased agonism, which means that when a molecule binds to a receptor, it biases the receptor towards a certain pathway because there's more than one pathway that can occur. And that's something we have yet to fully explore with the cannabinoids. And now I'm all off kilter. So let's finish. Sorry.
Okay, you want to just click through to the...
Yeah, we'll do the last one.
Okay, pretty much what we're talking about here is these specific benefits that are linked to more chemodiverse products because they have multi-target and multi-system modifications. These compounds not only interact and alter our endocannabinoid system, we know that some of the compounds in cannabis can also interact and modulate other systems in our body, such as the serotonin system, the dopamine system. There are plenty and things we don't even understand about what the endocannabinoid system controls, but it is involved in every single organ, every single process in our body. So the potential of cannabis medicine and chemodiverse products is unlimited. The question is, how do we understand it better? What tools can we use? What approaches can we take? Who can we talk to? What can we learn from you guys to understand better exactly what is happening with these different products?
Yeah, and this approach, the chemodiversity approach, is something that Riley and I are really passionate about, and it's the reason why our company does the research that we do. And it's this approach to illness. It's this approach to, let's not even say chronic illness, maybe it's like chronic wellness. It's this approach to our quality of life and the balance of our bodies, having to do with this multi-system, multi-target, multi-molecule chemodiverse approach to our wellness. And I just want to throw out there before we end that we are aware that there is tons of research out there on negative effects of cannabis smoke and other smoke, tobacco smoke, and they get kind of mixed in there as like similar research. We are not saying that any of that doesn't exist. We're not saying that it's not true. We're not saying anything about that. Like, I believe that research is out there and that there's plenty of other resources for that. We just are spending our time trying to talk about the things that other people aren't talking about.
Yeah, we know most people prefer smoking. We are two people who prefer smoking. So instead of just telling people, hey, don't smoke, instead we're going to say, okay, why do so many people prefer smoking? What is happening here? It's not just the physical act of smoking. It does feel differently. So our goal is to understand that and communicate our findings with everyone, because we do believe there's a lot of unique things happening during the smoking and combustion process that could be beneficial to patients and people everywhere.
And so we probably can do like one question. But thank you all so much for listening and for being a part of it. And this is what we do. This is our team. And if you haven't checked out our podcast, please listen to our podcast. You want to hear more about our team. Episode 1.6 is about all of us and why we do what we do. And in terms of like making a product that is more close to the feeling of smoking, like that is the research that we're doing. And that is what we're trying to put out there in the universe. I'll have to say that we're not perfect, but we're probably closer out there than other extracts. Like we there's work to be done, like to make it exactly the same, but I don't know how we're going to bypass first pass metabolism. That's going to be
and thank you to all the people who have purchased the hemp smokenol product. It means a lot for you guys to support us and support our message, and giving feedback on it also matters a lot. That's the first product that we've released, and we have more coming, but we love to hear from you guys. So thank you to everyone who's purchased that.
Riley, this question I'm punting to you. Do different cannabinoids absorb differently when used topically?
Thanks for attending our webinar on pharmacology of smoke. We hope it gave you something to think about at your next sesh.
If this webinar enlightened you on the unique chemistry of cannabis smoke and you would like to try some of that in tincture form, go to www.profoundnaturals.com. That's all one word, www.profoundnaturals.com, to try some of our hemp smoke extract today. We will always be capping these webinars to 100 registrants so that we can have time to take questions at the end and facilitate a more intimate networking experience in the chat discussion, but we will also always be posting them as videos for free.
So if you want to be the first to know when we're hosting another webinar and want to be one of the first to sign up for it, make sure you follow us on Instagram at smokenol, and listen to our podcast at Smokin' Science on all major streaming platforms. We will announce events that we do together through these accounts before we announce anything on our personal accounts.
Thanks again for attending, and we hope to see you all at another Smokin' Science sesh.
Mad love, homies. Thanks for coming.